Hi, we meet again today to do a drill revision together. Okay, so today teacher would like to do revision C4 and CAM plants. Okay, let's look at the first essay. So, question number one, this essay contained 8 marks. Okay, the question is describe adaptations of C4 plants in the terrestrial land and significance of plants anatomy to minimize photorespiration. Okay, so the object in the question is C4 plants. So your answer is supposed to mention about first thing, C4 plants live in hot and dry areas since the question asks adaptations of C4 plants in the terrestrial land and significance of plants anatomy. So C4 plants live in hot and dry area, correct? Then therefore the plants are forced to close their stomata to prevent excessive loss of water, dehydration via transpiration in the evening means day at the day in the day right so you got another mark. Then to prevent the effect of photorespiration, C4 plants exhibit Crohn's anatomy in which C4 plants have a large bundle shift cells around the vascular bundle. So from this explanation, you will obtain 3 marks. Okay, because this explanation answer the question. The question about C4 plants in the terrestrial land. Because C4 plants live in hot and dry areas, so thus they will close their stomata to prevent excessive loss of water via transpiration in the day, in the evening. So to prevent and then significance of Crohn's anatomy to minimize photorespiration. We understand that to minimize the photorespiration, C4 plants exhibit Crohn's anatomy. Okay, and this Crohn's anatomy contain large bundle shift cells. Okay, then we continue. You should continue your answer with this. Okay, so you need to continue with to mention in C4 plants what happened because they ask you the question asks you to minimize photorespiration, so you need to explain CO2 bound into phosphoenol pyruvate PEP in the mesophyll. Yes, in the C4 plants, CO2 bound into PEP in the mesophyll. So the CO2 then release into the bundle shift cells so that CO2 enters the Calvin cycle. So here you precisely you mention that CO2 bound into the PEP in the mesophyll, then later at another place in the bundle shift cells, CO2 enters the Calvin cycle. So the major function, okay, then you need to continue your answer with function of mesophyll cells for C4 plant is to pump CO2 into bundle shift cells. Okay, the significance is to keeping the CO2 concentration in the bundle shift cells always high enough for Rubisco to fix CO2 rather than oxygen, because the bundle shift cells is already away from the mesophyll, so the CO2 concentration is always enough for Rubisco to fix it. Okay, thus it minimizes photorespiration and maximizes sugar production. So here you will obtain 8 marks. Okay, if you answer what the question asks you. Okay, settle with the first essay. Now let's look at another essay. Okay, before that, okay, distribution of C4 plants. You need to know that C4 plants are abundant in hot climate, tropical or subtropical areas. Examples of C4 plants: maize, rye, grasses, and sugarcane, like in this picture, because their optimum temperature for C4 plants are above 25 degrees. Okay, we continue with second question. Draw Crohn's anatomy in C4 plants. So usually the labeling and the drawing just obtain 3 marks. Okay, let's say you need to draw quickly, a quick draw, you just draw the surface here where contain the label, then you need to label chloroplast inside in your drawing. Alizate mesophyll cell, okay, alizate mesophyll cell, and bundle shift cells. So at least your drawing need to have 3 labels, and the important labels are chloroplast, mesophyll cell, and bundle shift cell. Okay, you can draw this one quickly. All right, so you will obtain 3 marks. Okay, then we continue with the third question and the last essay for revisions today. Okay, for today revision. So explain H and slack pathway. H slack pathway. Obtain 8 marks from your answer. And before you start to write your answer, you need to have this diagram in your mind, that you need to memorize back that Hash slack pathway has a special separation of steps, mean they occur at the 2 places. First one in the mesophyll cell, mostly the main purpose, the process for carbon fixation in the mesophyll cell. So then carbon dioxide or CO2 released from the organic acid into the Calvin cycle in the bundle shift cell. So organic acids release CO2 to Calvin cycle. So you need to explain, all right, the details, step 1, 2, 3, 1 to 3 in the mesophyll cell, and then step 4, 6, step 4, 5, 6, 7, and 8, 4, 5, 6, 7 in the bounded shift cells, and the last one return back in the mesophyll cell. Okay, we just go through. Okay, here in mesophyll cell, please mention this in your essay. To answer this question, you need to mention the location of the process hash slack pathway. Okay, first stage, carbon dioxide is fixed by PEP in the chloroplast of the mesophyll cells. So the first, the receiver, a CO2 receiver in the hash slack pathway is a PEP. And then second stage, PEP after receive CO2, it becomes oxaloacetate, a 4-carbon compound. All right, so a 4-carbon compound formed as the first product of photosynthesis. So that's why the plants got their name C4 plants, because a 4-carbon compound is formed as the first product of photosynthesis. Then you need to explain that enzyme involved in this reaction is PEP carboxylase. Okay, also it is only 8 marks, but I strongly suggest that you need to mention the enzyme involved at the second stage. Then third stage, that require you to write oxaloacetate is reduced to malate, which will enter the bundle shift cells. And this require an ADPH2. So here, And ADPH2 were consumed, sorry, yes, was consumed to produce malate. Then in bundle shift cells, stage 4, stage 4 happened in the bundle shift cell. So at stage 4 in bundle shift cells, malate is oxidized into pyruvate and carbon dioxide. Okay, then stage 5. Okay, now NADP plus, right, reduced to NADPH2 so that malate 4 carbon will, malate is oxidized into pyruvate while NADP plus reduced to NADPH2, right? So and malate being oxidized and ADP plus being reduced, okay, and to produce or to form pyruvate 3 carbon and carbon dioxide. At stage 5, carbon dioxide accumulates in the cytoplasm of the where bundle shift cells and creates a high concentration of carbon dioxide in the bundle shift cells. At stage 6, that you need to write, carbon dioxide will enter the Calvin cycle and join RUBP to become 3-phosphoglycerate PGA. So the same thing, the Calvin cycle process is the same as C3 plants in order to synthesize organic compounds. Then pyruvate will return to the mesophyll cells by diffusion. So at stage 8, it happened in mesophyll cells. Pyruvate is converted into PEP again by consume 1 ATP via phosphorylation. All right, so ATP will become ADP. All right, and then pyruvate will form PEP in the H-slack pathway again. All right, so that's all. I hope you can write 8 stages of the H-slack pathways and obtain 8 marks. Okay, that's all for today. So to my students, please submit your essays to me. Okay, so okay, hope you can manage to write a good and factual essays to get a full marks in examination. Okay, bye. See you again.